What's up everybody? So in this video we're going to be talking about antibiotics. This is the last video in the C3.2 thing that we need to cover, right? So there are 4 videos that I made, and the biggest one was this one, Defense Against Infectious Disease. A lot of super important things were covered here. I also made a video on ABO and RH blood groups, and I made one as well on zoonotic diseases, and now we have this last one, antibiotics. This is all the videos you'll need for C3.2. So let's just get started. So a lot of people will get confused between these two words, antibodies versus antibiotics. In this video, we're talking about antibiotics, but we're also going to mention antibodies because that will make antibiotics, the difference between these two, much more clear for you. So what are antibodies? So antibodies is what I talked about in the other video, right? It's what our immune system normally does to fight off a pathogen, right? So if we have a pathogen, our B cells will recognize them, differentiate into plasma cells, and these plasma cells will make these antibodies, and these antibodies will attack that particular pathogen and destroy it through many ways, right? Now this pathogen can be any kind of pathogen. It can be what? A virus, a bacteria, a fungi, a parasite, any pathogen under the sun. Okay, because our immune system just has to recognize that pathogen and make this antibody. That's it, right? That's how it works. If our body works, we can do it. So I'm gonna quickly show you some words here because why not? So these things, I want to put them here because I just said a bunch of words, but I want to write them down so it's clear and you can remember them far more easily. So antibodies are made naturally by your lymphocytes, your immune system, against pathogens that invaded your body. Perfect. Antibodies can kill various types of pathogens, as I just mentioned. Antibodies are specific, meant to kill a specific pathogen. So right, you have many B cells. This is covered in that other video. So you have many B cells. Each B cell is destined to make one kind of antibody. Only one kind. Notice, they're all the same color here because this B cell is only meant to make this specific kind. Another B cell will be destined to make another kind. So you have so many B cells, and each B cell makes one specific kind of antibody because this specific antibody is meant for only this specific pathogen. This antibody cannot kill any other pathogen. It is meant for specifically this specific kind of specific bacteria. You see what I mean? So antibodies are specific. They're only meant for one specific pathogen. Did I say specific enough times? I think so. So antibodies bind to this antigen, right? This, again, if you don't get this, make sure to watch the other video. They bind to this antigen and kill them through various mechanisms. Okay, that's antibodies for you. So to understand antibiotics, right, they sound very similar. You need to understand this. So we know life comes from a common ancestor, and over time, 3 categories of life was kind of formed, right? 3 categories: eukarya, archaea, and bacteria. Eukarya, these organisms, include organisms like me and you, like the elephant outside, like the plant by your window. All these organisms are eukaryotic cells. We're made up of eukaryotic cells, right? Many of these eukaryotic cells. This is the one specific kind of cell. The other 2 categories, their cells are kind of different. They're made up of cells that are slightly different from the eukaryotic cells, and they are, in fact, prokaryotic cells. And don't worry too much about these differences. There's a whole video on eukaryotic and prokaryotic cells in another chapter, so don't get too caught up on this. I'm just trying to get a point across here. So the point is that I'm getting across is that there are 2 big categories of cells: prokaryotic cells and eukaryotic cells, and they're different based on their structure. They're slightly different. But importantly, humans fall in this category of eukaryotic cells, and bacteria fall in prokaryotic cells. Bear this in mind, very important. Okay. So if this is our prokaryotic cell, our bacteria, right? This is one kind of pathogen only one kind. It's not a virus, it's not a fungi, it's not a parasite, it's a bacteria. It's one kind of pathogen, a prokaryotic cell. And it's got a certain structure. Don't worry about this. I'm just trying to get a point across. It's got a lot of structures, right? This prokaryotic cell. Now, antibiotics is little medications. Don't worry, I'll put the words down on the next slide, so don't worry about writing anything down. All the words will be there. So antibiotics is little molecules made by scientists, little chemicals that are going to target the bacterial functions. For example, the bacteria has ribosomes. Ribosomes make proteins. So if the scientists can make a kind of chemical that can target this ribosome, it can kind of kill this bacteria, because without the ribosomes working properly, the cell cannot make proteins, right? The ribosomes make proteins, and proteins are needed for pretty much every function in a cell. So in that way, you can kill them. Antibiotics can also be a molecule that's targeting the cell wall. If you can prevent the cell wall from being healthy or being formed, then you can kill the bacteria. So in that way, a brief intro to antibiotics is that it's a little molecule that is against bacteria only. Bacteria only, not any of the other pathogens, just bacteria. And it works by inhibiting the metabolism. For example, metabolism is just any kind of reaction. So ribosomes is involved in reactions making proteins. So if you can inhibit this kind of reaction, then you're killing the cell. If you're inhibiting this function, which is the cell wall, you're killing the cell, okay? So that's a vague intro. So we're gonna get now the words down and everything will be clear. So let's get some words down. Oh yeah, here, a big brain tip for you. If you get confused between antibodies and antibiotics, antibodies is made by your body. Antibiotics sounds like robotic, so you can think it's unnatural or made by scientists. It's not from your own body, okay? Great. It's medication. It's what you take from the doctor when you're sick. Okay, so antibiotics are made artificially in a lab or made by microorganisms, and then scientists essentially steal it from the organisms that makes it. Let me give you an example. So either scientists can specifically make this molecule that's going to target the ribosomes, like I just mentioned, and that way they'll kill the cell. But sometimes they don't even need to make them. Sometimes this can happen. Say there's a bacteria that infects you and infects the horse. Your body somehow is useless and cannot make antibodies to fight off this specific infection. You're going to die from it. But this horse got infected by the same specific bacteria, and but it somehow can make little antibodies, can make little molecules that can fight it off. So in this scenario, the scientists will take the blood sample from the horse and isolate this little molecule that the horse was able to make, that a human was not able to make, and it'll kind of synthesize and duplicate this molecule many, many times and give it to you, and then you're saved. So you were technically saved by the horse. So that's another example of how antibiotics can be made. So not only synthesized by a scientist, but also taken from other organisms. Okay, other organisms. Okay, great. Next. Okay, very important. Antibiotics kill only bacteria, not viruses or anything else. I cannot overemphasize this. Only bacteria. Okay, kind of think of it like antibacteria. Only bacteria, not viruses or anything else like the antibodies were able to. Okay, antibiotics are non-specific. So remember, antibodies were specific. That specific antibody could only kill one kind of specific pathogen, whereas antibiotics, it is general medication, like as I just said. So the scientists will make a medication against ribosomes, these 70S ribosomes. So all bacteria have the 70S ribosomes, so that medication will not only work for this kind of bacteria, but it will work for every kind of bacteria, because all bacteria have 70S ribosomes. All bacteria have a cell wall. So in that way, antibiotics, unlike antibodies, is non-specific. One kind of medication could work for almost every kind of bacteria, okay? Now, last one here. Antibiotics don't bind an antigen, right? They bind and inhibit some kind of thing inside the bacteria, some kind of process that's going on inside the bacteria, some kind of metabolic process. Okay, so that's also different from antibodies. Okay, now the one question is, why do antibiotics, when you take an antibiotic, right, you take this medication, it's meant to kill cells. Why does it not kill your own cells? And why does it only kill these bacteria? How does that work? The reason being because fundamentally, as you know, prokaryotic cells are different from eukaryotic cells. The structures are different. The ribosomes in prokaryotic cells are 70S, right? We can see from this one. The eukaryotic cells are 80S, so the ribosomes are different. This antibiotic that was made is meant to target 70S ribosomes. So it's going to kill prokaryotic cells, but not eukaryotic cells. This medication is utterly useless for 80S ribosomes. On the other hand, say we have a medication for the cell wall. Does a eukaryotic cell have a cell wall? No. So there's no way that this medication can damage your own cells. It can only damage this prokaryotic cell. So that's very interesting how it works. So you will not get damaged by antibiotics yourself. And the other point I want to say here is do not take antibiotics for a viral infection. Sometimes you're sick, don't just take antibiotics. Don't just do that. You may be infected by a virus and it'll be useless. And I'll talk about that later in this video. Okay, that later just arrived, by the way. We're talking about viruses. So a virus, okay, a virus is dead, okay? A prokaryotic cell is alive. A virus is dead. It has no metabolism. Antibodies do not work. I just told you that. Okay, why? What's happening here? Why not? Why can't we just take this thing to kill viruses? Because viruses work like this. If this is our virus and this is a cell in our body or some bacteria, anyways, any living cell, a virus cannot survive by itself. It only survives by using a living cell. For example here, if we say this is a virus and this is its DNA, what it does is it attaches to a specific cell. It injects its DNA. Okay, so it injects its DNA. Give me a second. Okay, it injects its DNA. Once the DNA is inside, remember, DNA is genetic material. It's an instruction manual. This cell will not know if this DNA is its own DNA or if it's this virus's DNA. It won't know. It will just know it's DNA and it's going to do what it says. So it starts doing what it says. It uses the ribosomes. I didn't put all the ribosomes and stuff in here, all the organelles and all that, but whatever. It uses this DNA and it reads it and it does what it says. So it's going to make a virus. And so after a while, it made a full virus, and then eventually it's going to get so full, it's going to explode and release all of these viruses. So you can see a virus actually doesn't have any of the things, doesn't have ribosomes, it doesn't have those kind of things, those kind of organelles or enzymes inside itself to be able to duplicate itself. It needs to use a living cell to do so. So antibiotics are useless because antibiotics target ribosomes and cell walls. Viruses have none of this. They're not even alive. Okay, so it's not going to work. You need to use other things to target viruses. For example, antiviral medication. What do you think antiviral medication will do? Okay, a virus attaches to the cell. So one way to prevent a viral infection. So the only way a virus stays alive is by attaching to you and making itself replicate. So if it cannot attach to you in the first place, isn't that a good medication? Yes. One of the medications, antiviral medications, suppress viruses' ability to infect you, suppress the ability of them attaching to you and injecting stuff into you. So if you can inhibit this step, that's already a very good medication. Okay, so that's kind of the idea you're targeting. It's different from the prokaryotic cell. So understand that viruses are dead. They do not have metabolism, so these antibiotics will not work. There is no ribosomes to target. There is no cell wall to target. Okay, it just won't work. Okay, so you need to use other kinds of medication. Okay, great. So here's the table I promised you that's comparing antibodies and antibiotics side by side. So I hope this one is useful for you because you know, I put effort to make this just for you, you know. Okay, now we're getting near the end here. So I want to talk a little bit more about antibiotics, right? Since that's the topic of this video and you need to know this stuff. In science, we can have certain scientists, let's say, and they can do experiments. So when you get sick, let's have Mr. Beast here. When he gets sick, he's terribly sick, right? His body is not coping. He needs to plan more videos. He cannot be sick right now. He needs to get healthy. He needs to get antibiotics, right? So what's he gonna do? So scientists have pretty good ways of making sure you get the right antibiotic. Let me show you. So here we have a little petri dish, and it's got in here fluid it's perfect for bacteria. Like there's a lot of sugar in here, there's a lot of nutrients in here for these bacteria to survive, okay? So here's the dish, here's a lot of bacteria surviving, okay? Now, what they do is, so this, for example, could be the bacteria that Mr. Beast was infected by. So a sample was taken, put here to be able to see, to be able to grow them. Why do we want to grow them? Why does the scientist want to grow the bacteria he's infected by? They took a sample, they put it in this petri dish, they grow it. Because they want to do this. They want to put medication. So on here, they put a certain kind of antibiotic, and at the bottom, they put a certain kind of antibiotic because they want to see which ones work, which ones don't. And you can see this antibiotic, the whole area around it, all the bacteria died. There was no growth. So this antibiotic is effective for the specific kind of bacteria Mr. Beast was infected by. Whereas this other one, which is also an antibiotic, was not effective. So in this way, scientists like to do a culture. They like to culture your bacteria. They take a sample, they culture it, and they kind of test different kinds of antibiotics to see which one works the best. And then they will give that one to Mr. Beast and not that one, even though they are both antibiotics. So we can see in this one, these bacteria were sensitive. Sensitive means they get affected by very easily. They get killed very easily by this bacteria, by this antibiotic, but it was resistant, meaning they don't get affected by this antibiotic. Okay, so here's a real image. So you can see if this, let's say this whole thing was done for Mr. Beast. We got this one here. We got all these little circles. They are little antibiotics, antibiotic pills. And you can see here, this whole greenish area, that's all the bacteria growing. So you can see around some of these pills, there was a lot of death. No bacteria was able to grow. But then around some of these other ones, bacteria were able to grow. So which ones were effective? All of the ones with a huge space of no bacteria was effective. And all of the ones that had these green growths around it, all those areas are bacteria, which means they did not work. So let me label this for you. So we can say these areas all have bacteria growth. This area will have no growth, which means this one was very effective. There's no growth around it. Everywhere around this bacteria, antibiotic, all the bacteria died. Whereas this one, all the bacteria survived. So that one's useless. So these are all antibiotics. So useless, useless, useless. The rest were useful. And then they will see which ones were more useful. Because look at this one. It's got a small ring where there was no bacteria, but this one's got a big ring. So maybe it's more useful in fighting this bacteria. So I wanted to show you this because this is a real, you need to know this. And this is a real application of these antibiotics and how they use them sometimes to treat people. Because now they know this one is going to work for Mr. Beast. He can go back and make more videos, make a lot more money, a lot more views, and he'll be all fine, right? He can make money. If they gave him this one, he would not be all right, and then he would be sick for a while more. Okay. Okay, so we need to now understand something. We start here. Sometimes you can give bacteria to you can give antibiotics to certain bacterial infection and it will go away. But sometimes over time, these bacteria can start having mutations in their DNA. So, for example, you can see this one here, their DNA changed a bit. They had a mutation. So they're not exactly the same as these other bacteria anymore. So when you give now antibiotics, all these blue ones will be killed. But because this one had a mutation, it will survive. And if it survives, it will duplicate and it will grow. So we call this antibiotic resistant bacteria. Because it was mutating over time, because it takes time, this new strand was formed, and this one was resistant to this antibiotics. And now it replicates, and now we have a problem because the antibiotics don't work and you're still sick. So there's a specific kind of infection you need to know about that is like this, and it's called methicillin-resistant Staphylococcus aureus. So Staphylococcus aureus is a science name for a kind of bacterial infection. It's a kind of bacteria. And when you get infected by this, you can normally just take medication, antibiotics, and it'll go away. But there's this kind of mutation that can happen that makes these bacteria resistant to a certain antibiotic. And this antibiotic that they become resistant to is called methicillin. So therefore, they call these resistant bacteria methicillin-resistant Staphylococcus aureus. So we can pretend this was Staphylococcus aureus. Over time, there was this mutation. Now they are resistant to this antibiotic, so we call them methicillin-resistant Staphylococcus aureus. And they are really difficult to treat because now you got to find new antibiotics that are stronger and maybe more risky to use. And it may take even longer to remove this bacteria. So this concept is important to understand. You need to understand that certain bacteria over time can develop mutation, and that can make them resistant to antibiotics. So how can we prevent this problem? Because won't this always happen? There's a way you can kind of minimize and prevent this. Let me show you. The normal course of disease goes like this. You get infected by a bacteria, you develop symptoms, you go to the doctor, they give you antibiotics, you take them, your symptoms go away, and eventually you have a cure, right? And then you stop your antibiotics. So a lot of people have a problem. They get antibiotics, okay? This may be you, maybe you're guilty. They get infected, they get symptoms, they get antibiotics, they end their symptoms, and they stop their antibiotics. Even though they have more, they stop it. Now guess what happens if you stop antibiotics? Maybe you took your antibiotics and you killed maybe you killed most of the bacteria, but one remained. And then this one remaining over time, because you didn't kill it, because you stopped taking antibiotics, because maybe, like, let me reiterate this. Maybe if you have 5 bacteria and you remove 4 because you took antibiotics, maybe you feel good now, your symptoms went away, but you still have this one left. And this one can, over time, if you don't get rid of it, mutate and develop this mutation. And now, because you stopped taking antibiotics, this one was able to mutate and now duplicate and become resistant to the antibiotics. And now you have a problem. So you should not end your antibiotics until you're cured, until the doctor, until you finish the prescription that the doctor gave you. Otherwise, you may have eliminated most of the bacteria, but this 1 or 2 remained. And despite the fact that you have no symptoms, these 1 or 2 that remain can mutate. And if they mutate, like this one, then you have a problem. And then you will be sick and you need to take more antibiotics, do more tests and all that. So the point is, do not end your antibiotics early. Wait for them after you're cured, after the doctor said so. And then, yeah, so that's the point I'm trying to make. So do not stop your antibiotics. Do not stop it. Continue it until you're done. And also don't, this is a kind of a farm link. Don't give it to animals. As a farmer, you don't want to give, sometimes naturally farmers want to give antibiotics to their animals so they don't get sick. Don't give too many if you're a farmer to your animals, because this can also cause this kind of situation to happen, making these resistant bacteria. Now, if this cow infects you, you have now this annoying resistant one instead of this nice one that's sensitive. So that's also a thing. We want to avoid forming all of these resistant ones. So let's do 3 IB questions and that'll be it for this video. So why is penicillin not used in the treatment of HIV? So HIV is a virus, as you can see, human immunodeficiency virus, and it causes AIDS in the long run. Penicillin is a bacteria. So why is this bacterial medication, penicillin is an antibiotic. So why is this antibiotic not used in the treatment of HIV? Because I just told you, you cannot use antibiotics for virus infections. So let's see. HIV patients may be allergic to penicillin. Nope, that's not the reason. Penicillin does not affect viruses. Great. Penicillin affects bacteria only. Penicillin affects helper T. Nope. Penicillin causes antibiotic resistance. Nope. Why are antibiotics effective against bacteria? They can produce specific antibodies. No. They can engulf foreign matter. No. They can block specific metabolic pathways. Yes, such as ribosomes, such as cell walls. Yep, they can do this. They can act as a vaccine. No, they cannot. What is the reason for antibiotics not damaging human cells? We talked about this one, right? So the dose is too small to be harmful. Nope, that's not true. The dose is taken for only a short time. Nope. Antibiotics occur naturally in humans. No, they don't occur naturally. Human metabolism is different from bacterial metabolism. Yes, the cells are different. So their metabolism, their reactions, their organelles are all different. Therefore, the antibiotics will not work against the eukaryotic cells, only the prokaryotic cells. So that is it for this video. We covered everything about antibiotics. There's a lot of things there. Hopefully they all make sense and you learned something in this video. And I'll see you in the next one.